Hello and welcome to another video discussion. And in this video, we'll talk about biotechnology. Although several discoveries have been made in biology and technology over the years, which ultimately led to its development, the term biotechnology is believed to be first coined by Hungarian agricultural engineer Karl Erecki in 1919. He built a slaughterhouse and a farm for thousands of pigs and produced over 100,000 pigs in a year. According to him, biotechnology was the process of upgrading the raw materials biologically into socially useful products. For that, he is regarded by some as the founding father of biotechnology. Biotechnology, in definition, is the science of using living organisms or the products of living organisms for human benefit or consumption. Today, biotechnology is defined by different organizations in several different ways. In the recent years, the scope of biotechnology has expanded. Biotechnology is the use of living organisms and systems to manufacture useful products or to perform an industrial task. Biotechnology encompasses both traditional or what we call classical biotechnology and the modern technology. But without the traditional biotech, there won't be any modern biotechnology. The diversity of microorganisms and development of genetics expanded the potential of traditional biotechnology and ultimately led to the development of modern biotechnology. But what is traditional or classical biotechnology? Traditional biotechnology refers to the traditional techniques of using living organisms to yield new products or modify foods or other useful products for human use. The early examples of biotechnology include breeding animals and crops to make cheese, yogurt, bread, beer, and wine. Some traditional techniques such as selective breeding, hybridization, and mutagenesis are used in current applications of biotechnology. Other technologies include fermentation, selective breeding, food processing, tissue culture, and more. The earliest example of biotechnology is the domestication of plants and animals. Domestication began over 10,000 years ago when our ancestors started keeping plants as a reliable source of food. Rice, barley, and wheat were among the first domesticated plants. Wild animals were tamed to provide milk or meat or help with plowing or guarding the farm. The dog, sheep, and goat are thought to be among the first animals that were domesticated. Genetic engineering, artificial selection, antibiotic production, and cell culture are current topics of study in biotechnology. However, humans have used prokaryotes before the term biotechnology was even coined. Some of the products are as simple as cheese, bread, wine, beer, and yogurt, which employ both bacteria and other microbes such as yeast. Egyptians used yeasts to bake leavened bread. Bread may be leavened by naturally occurring microbes, chemicals, industrially produced yeast, or high-pressure aeration, which creates the gas bubbles that fluffs up the bread. Yeasts are single-celled fungi. As fungi, they are related to other fungi that people are more familiar with, including edible mushrooms available at the supermarket, common baker's yeast used to leaven bread, molds that ripen blue cheese, and the molds that produce antibiotics for medical and veterinary use. The scientific name for the yeast that bakers use is Saccharomyces cerevisiae, or sugar-eating fungus. The process alcoholic fermentation produces useful end products, carbon dioxide and ethyl alcohol. These end products are released by the yeast cells into the surrounding liquid in the dough. In bread baking, when yeast ferments the sugars available from the flour and or from added sugar, the carbon dioxide gas cannot escape because the dough is elastic and stretchable. As a result of this expanding gas, the dough inflates or rises. Kaya nga yung mga leavened breads ay ang lambot pag kinakain natin, di ba? Thus, the term yeast leaven breads was added to the vocabulary of the world of baking. The ethyl alcohol produced during fermentation naman produced the typical flavor and aroma of yeast leaven breads. That's why they say the bread is more delicious if you will use yeast. Cheese production began around 4,000 to 7,000 years ago when humans began to breed animals and process their milk. Fermentation, in this case, preserves nutrients because milk will spoil relatively quickly. But when processed as cheese, it is more stable. A required step in cheese making is separating the milk into solid curds and liquid whey, and this usually is done by acidifying the milk and adding rennet. The acidification can be accomplished directly by the addition of an acid like vinegar, but usually starter bacteria are employed instead. These starter bacteria convert milk sugars into lactic acid. The same bacteria also play a large role in the eventual flavor of aged cheeses. And most cheeses are made with starter bacteria from the Lactococci, Lactobacilli, or Streptococci families. As a cheese ages, microbes and enzymes transform texture and intensify flavor. This transformation is largely a result of the breakdown of casein proteins and milk fat into a complex mix of amino acids. And do you know that some cheeses have additional bacteria or molds intentionally introduced before or during aging? In traditional cheese making, these microbes might already be present in the aging room. They are simply allowed to settle and grow on the stored cheeses. And more often today, prepared cultures are used, giving more consistent results and putting fewer constraints on the environment where the cheese ages. Records of brewing beer date back about 6,000 years to the Sumerians. Evidence indicates that the Sumerians discovered fermentation by chance. Wine has been produced for about 4,500 years, and the production of beer and wine use microbes, including both yeast and bacteria, to produce ethanol during fermentation, as well as provide flavor to the beverage. According to the Algae Industry magazine, early Spanish writings document the Aztecs harvesting green algae from lakes by boat, then drying it in the sun to produce a green cake. Spirulina has been hailed as a superfood due to its high concentration of protein, vitamins, minerals, nutrients, carotenoids, and antioxidants. Another early process of biotechnology is what we call selective breeding. Selective breeding involves choosing parents with particular characteristics to breed together and produce offspring with more desirable characteristics. Selective breeding aims to adapt an organism's characteristics in a way that is desirable to the humans that breed them. And humans have selectively bred plants and animals for thousands of years, including crop plants with beer yields, ornamental plants with particular flower shapes and colors, farm animals that produce more better quality meat or wool, dogs with particular physiques and temperaments suited to do jobs like herd sheep or collect peasants. We have now a modern way of producing vaccines and antibiotics, but in their early development, they're still under traditional biotechnology. Antibiotics are biotechnological products that inhibit bacterial growth or kill bacteria. They are naturally produced by microorganisms, such as fungi, to attain an advantage over bacterial populations. Antibiotics are produced on a large scale by cultivating and manipulating fungal cells. Now, noon kasi, pag nilagnat ka, parang delikado na yung buhay mo, di ba? But because of the invention of the penicillin by Alexander Fleming, no, by the use of these microorganisms called fungi to kill bacteria, then yung lagnat ay naging typical na sakit na lang na pwedeng gamutin. Now, another one is the vaccination. Traditional vaccination strategies used weakened or inactive forms of microorganisms then to mount the initial immune response. So, it's still under traditional biotechnology. Pero syempre, yung mga modern way of producing vaccines ngayon, kagaya sa COVID-19 vaccines na meron tayo, mas modern na yung way. So, yung mga yon ay under na ng modern biotechnology. But what is modern biotechnology? Modern biotechnology involves the use of GE techniques such as recombinant DNA, functional and structural genomics, DNA diagnostic probes, and other methods for genetic modification. The development of recombinant DNA technology has marked the beginning of so-called modern biotechnology. Modern biotech has contributed significantly to enhancing our knowledge of biological systems. Modern biotechnology techniques are currently being used in many areas such as food, agriculture, forestry, healthcare, environment, minerals, and industrial processes to develop new products and processes. And one of the most extensive applications of modern biotech is in agriculture. Recombinant DNA techniques and mutagenesis are used to develop plants with novel traits. Biotech diagnostics have been used to detect a wide variety of diseases and genetic conditions. DNA cloning is one of the important processes in modern biotechnology today. This is a molecular biology technique that makes many identical copies of a piece of DNA, such as gene. But what is the point of making this? In some cases, we need lots of DNA copies to conduct experiments or build new plasmids. In other cases, the piece of DNA encodes a useful protein, and the bacteria are used as factories to make the protein. For instance, the human insulin gene is expressed in E. coli bacteria to make insulin used by diabetics. DNA molecules built through cloning techniques are used for many purposes in molecular biology. A short list of examples includes what we call biopharmaceuticals. This is the DNA cloning where it can be used to make human proteins with biomedical applications such as the insulin mentioned above. And other examples of recombinant proteins include human growth hormone, which is given to patients who are unable to synthesize the hormone, and tissue plasminogen activator or TPA, which is used to treat strokes and prevent blood clots. Recombinant proteins like these are often made in bacteria. The other one is what we call gene therapy. In some genetic disorders kasi, patients lack the functional form of a particular gene. Now, gene therapy attempts to provide a normal copy of the gene to the cells of a patient's body. For example, DNA cloning was used to build plasmids containing a normal version of the gene that's non-functional in a cystic fibrosis patient. When the plasmids were delivered to the lungs of cystic fibrosis patients, lung function deteriorated less quickly. Okay. Now, we have also what we call gene analysis. In basic research labs, biologists often use DNA cloning to build artificial, recombinant versions of genes that help them understand how normal genes in an organism function. These are just a few examples of how DNA cloning is used in biology today. DNA cloning is a very common technique that is used in a huge variety of molecular biology applications. In the previous slide, I spoke about genetic engineering or GE. Genetic engineering or GE is the alteration of an organism's genotype using recombinant DNA technology, and that is to modify an organism's DNA to achieve desirable traits. So we are literally manipulating our genes to modify some traits that we don't like or what we don't need in our body, or to prevent any diseases that are hereditary on our part. Now, how about GMOs? The organism that receives the recombinant DNA is called a genetically modified organism or GMO. If the foreign DNA that is introduced comes from a different species, the host organism is called transgenic. Bacteria, plants, and animals have been genetically modified since the early 1970s for academic, medical, agricultural, and industrial purposes. In the US, GMOs such as Roundup Ready soybeans and borer-resistant corn are part of many common processed foods. Transgenic modification, adding recombinant DNA to a species, has led to the expression of desirable genes in plants and animals. Now, although several recombinant proteins used in medicine are successfully produced in bacteria, some proteins require a eukaryotic animal host for proper processing. Now, for this reason, the desired genes are cloned and expressed in animals such as sheep, goats, and chickens. Animals that have been modified or expressed in recombinant DNA are called transgenic animals. Several human proteins are expressed in the milk of transgenic sheep and goats, while others are expressed in the eggs of chickens. Mice have been used extensively for expressing and studying the effects of recombinant genes and mutations. Manipulating the DNA of plants has helped to create desirable traits such as disease resistance, herbicide and pesticide resistance, better nutritional value, and better shelf life. Plants are the most important source of food for the human population. Farmers develop ways to select for plant varieties with the desirable traits long before modern day biotechnology practices were established. Plants that have received recombinant DNA from other species are called transgenic plants. Because foreign genes can spread to other species in the environment, extensive testing is required to ensure ecological stability. Staples like corn, potatoes, and tomatoes were the first crop plants to be genetically engineered. Now, if you want to know more about the specific examples of these transgenic organisms that were developed in the recent years, the following pages in our interactive book will provide this information.